My name is Tiffany Smith. I'm an MB3 major at Towson University, and today I'll be presenting on protein structure. There are four parts to protein structure. There's a primary structure, there's two different types of secondary structures, there's a tertiary structure and a quaternary structure. So basically a primary structure is just a single chain polypeptide. Secondary structure is made up of the connection between many primary structures, and secondary goes towards tertiary, meaning that these become engulfed together and it makes quaternary structure. So it goes, the quaternary structure is just basically more like approximately based on this, four tertiary structures. Now we call this a beta pleated sheet. It has a polypeptide backbone, and it has different atoms along it. This is an alpha helix. Same thing, they have the similar bonds and stuff. Before I go into detail, I will just explain the bonds to you. Okay, here we have an oxygen and a hydrogen. So that's a hydrogen bond. We have a sulfur bonded to another sulfur, so that's a disulfide bond. And we have an NH3 plus and an O minus bonding together. They're both charged, so they're ionic bonds. Now we have a CH3 to a CH3, which has no oxygen, so that means it's a van der Waals bond, or in other words, a hydrophobic bond. Now in here, this bond is composed of H and C and CH3, CH2. It's just a very densely hydrophobic set of bonds, and so we'll call that a hydrophobic exclusion. Now, for hydrogen bonds, we can find them. There's some here. The red is oxygen and the white is hydrogen. So we have a hydrogen bond. We have a carbon to oxygen, a carboxyl bond. That's black to red. And over here you can see another carboxyl bond and a hydrogen bond. Carbon to carbon is like pretty much the same. So I guess that's how they became secondary. And now down in tertiary, we can see alpha helices and beta pleated sheets together to form tertiary, and it's in a 3D structure. So now it brings across one tertiary structure, and then if you have like a couple more, then it's all together be quaternary. These are mostly weaker bonds because it's made up of a lot of different hydrogen bonds. So and hydrogen bonds are not really that strong. The strongest bonds are like covalent bonds, like the disulfide bond, because they're equally sharing, so they will be much stronger than, even a charged one. The terns are in R groups mostly. You see some R groups along here. The R groups are usually like hydrogen, like it could be just a hydrogen, or it could be hydrogen bonded to oxygen, carbon double bonded to oxygen, things like that. So that's just like a placeholder. And one thing to confirm why it's a protein, we have hydrogen, this one, hydrogen bonded to a carbon. So that's the main hydrogen. And then we have NH, although they came together, so it lost H. So that would be the amino group, and here's the carboxyl group, and then the R group. Those are the main parts of protein to identify it. So that's how we can confirm that this is a legit protein structure. It's also evident in the rest as well. You can see not completely, but the bonds are there. We have a N here and a H. So yeah, each turn is like a R group. Yeah, that's it. Have a good day.